Following multiple outbreaks in Africa, starting back in the 70s, in 2015, the WHO listed the Ebola virus disease as one of the top 10 emerging diseases. Today, the management of this contagious and deadly disease continues to pose challenges that only effective vaccination can stop. The clock is ticking, as new outbreaks continue to afflict the local populations and overwhelm the healthcare system. An innovative approach is needed. The development of candidate vaccines follows strict rules to demonstrate their safety and efficacy. This includes clinical testing in thousands of subjects to collect comprehensive data. The findings are peer-reviewed in scientific journals and are scrutinized by the authorities during the regulatory review process that is conditional to the official approval of the vaccine candidate. One of the greatest challenges is the demonstration that the candidate vaccine actually works by showing protection of the target group from clinical disease. This requires large, randomized clinical trials comparing the number of cases with disease among the vaccinated and the unvaccinated. While they get naturally infected, lower numbers of the diseased in the vaccinated group as compared to the non-vaccinated allows calculating the protective efficacy offered by the vaccine. However, for diseases like Ebola, the local circumstances make it difficult to perform such efficacy trials. Reasons for that include the unpredictable pattern of the epidemics, the sometimes brief duration of an outbreak, the inability of the local medical infrastructure to respond fast enough, and the interferences from social or political issues like mistrust or armed conflicts. In such circumstances, where the demonstration of vaccine efficacy in a human clinical trial is technically, logistically, or ethically not feasible, the US regulatory agency FDA allows that an immunological marker that correlates with protection from disease in a suitable animal model can be used to infer similar protection in humans. This is known as the FDA Animal Rule. The principle was also adopted by similar regulatory guidelines, including the European Medicines Agency's approval under exceptional circumstances. Janssen has developed a 2-dose vaccination against Ebola virus disease. The regimen consists of 1 dose of Zabdeno, followed by a dose of Mvabia. As an innovative alternative to a large clinical efficacy study in humans, Janssen has determined the protective effect of its vaccine candidate by comparing the immune response in humans to the immune response required for protection in a non-human primate model. For Ebola, a fully lethal Ebola virus challenge model in cynomolgus monkeys is the most relevant animal model, with similar Ebola virus disease symptoms as in humans, but with even more aggressive disease course with higher mortality. In that way, the model may even underestimate the potential effect of the vaccine in humans. Let's take a closer look at how the animal model was set up. A group of animals received the Janssen candidate Ebola vaccine regimen, while another group served as non-vaccinated controls. All animals were subsequently infected with the Ebola virus. The serum level of binding antibodies, as well as the survival of the animals in both groups, were evaluated. Binding antibodies induced by the vaccine regimen against the Ebola virus surface glycoprotein were identified as an immunological parameter correlating strongly with protection. At the clinical vaccine dose, 100% of the vaccinated animals were protected. Non-vaccinated animals did not raise antibodies and succumbed. To further examine the relationship between immune response and protection, lower doses of the vaccine were tested to draw a curve relating the level of antibodies to the survival rate. The relationship between the immune response and the survival probability in the non-human primates was assessed by logistic regression analysis. Vaccination in humans induced similarly strong anti-Ebola virus surface glycoprotein binding antibodies. By comparing the vaccine-induced antibody levels in humans with the antibody levels in the animals, a method called Immunobridging protection in humans could be inferred based on the observed relationship between antibody levels and the survival rate in the animals. The width of the confidence interval is a statistical expression of the uncertainty around the observation. To ensure that the observed result was sufficiently robust, It was agreed with the US and EU regulatory agencies that the lower limit of the 95% confidence interval should be at least 20%. The mean predicted survival probability in humans based on this model was found to be 53.4%, with the lower limit of the confidence interval at 36.7%. So the preset regulatory criteria were met. The robustness of these results was further confirmed by additional sensitivity analyses, repeating the calculations in multiple subpopulations in various geographies. Based on this immunobridging, the Janssen vaccine regimen is likely to protect humans against clinical Ebola virus disease. As the exact level of protection provided by the vaccine in humans can only be established in field efficacy trials, Janssen has committed to collect clinical efficacy information whenever feasible. The Janssen 2-dose vaccine regimen Zabdeno-Mvabia against Ebola virus disease was approved in Europe in July 2020, following demonstration of its quality, safety, and efficacy based on this innovative and robust immunobridging approach. Thanks to this immunobridging, Janssen was able to make the vaccine regimen available as an additional tool in the relentless fight against the Ebola virus.